this mRNA is released. This mRNA is released. This is the newly synthesized mRNA chain, mRNA molecule, which has a sequence of nucleotides complementary to the sequence of nucleotides in the sense strand. There are two things that you must remember here. Number one, I was telling you that after the mRNA is released, the two strands of DNA link up again and its original shape is restored. This may not happen always. The same sense strand may continue to provide code for synthesis of more molecules of the same mRNA. That means several chains of mRNA, can be synthesized from the same sense strand. This is one important feature. Second, RNA polymerase in eukaryotes is of three types. In prokaryotes, only one type of RNA polymerase is formed, but in eukaryotes, three types of RNA polymerase: RNA polymerase 1, RNA polymerase 1 specifically for the synthesis of ribosomal RNA, RNA polymerase 2, RNA polymerase 2 specifically for the synthesis of mRNA, and RNA polymerase 3 specifically for the synthesis of tRNA. So, mechanism is the same. Whether this RNA is going to be rRNA or mRNA or tRNA is decided by the enzyme involved: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Number 3, this entire process of transcription, occurs in the nucleus in eukaryote cells, but in prokaryotes no distinction, so everything happens in the cytoplasm. And another important situation is there is what is called as processing of RNA. Processing of RNA. The RNA that is synthesized like this, it may be ribosomal RNA, it may be messenger RNA, it may be transfer RNA. Most often messenger RNA is subjected to a processing in the nucleus before messenger RNA is released into cytoplasm. This processing is required because RNA contains not only expressing regions, sequence of nitrogen bases that function as codons, expressing regions. These expressing regions are called exons, as well as intervening regions. Intervening regions, sequence of some nitrogen bases that do not function as codons. These are called introns. Introns. So processing basically involves removal of introns, and joining of exons in a specific sequence. Which can be used for the process of translation. Then at the 5' end, some unusual nucleotides may be added. Some unusual nucleotides may be added at the 5' end. And this is called capping. At the 3' end, polyadenylate nucleotides, polyadenylate nucleotides may be added. This is called tailing. The removal of introns and joining of exons, this is called splicing. So after the formation of the mRNA chain, in the nucleus, the mRNA may be subjected to splicing, capping and tailing. Splicing is done to see that unwanted non-functional intervening regions are removed, whereas capping and tailing are done in order to protect the mRNA from getting dissolved within the nucleus or from getting acted upon by enzymes within the nucleus. So, for protecting the mRNA from being destroyed, capping and tailing is done, and for making the codons appear in a particular sequence, splicing can be done.